Welcome to the NanoCap podcast. I'm your host, Will Chernlin, and I'm excited to have you join me on this journey through the world of NanoCap companies. For the past decade, I've talked to thousands of business owners across various industries. It's been an enlightening experience revealing the vast tapestries of entrepreneurship and innovation that thrives beneath the surface of mainstream media coverage. Each week, I will interview different leaders of NanoCap publicly traded companies to learn about their history, what they do now, and their future. As a reminder, none of this information is financial advice, and it is all for entertainment purposes only. Let's start this week's episode. Hi, everyone. This week we have Dr. Marc Hedrick, CEO of Plus Therapeutics, stock ticker PSTV. Plus Therapeutics is a clinical stage pharmaceutical company developing innovative targeted radiotherapeutics for adult children with rare and difficult to treat cancer. Marc, thanks for coming on the podcast.
Hey, Will. No, my pleasure. Thank you.
I usually like to kind of start with people's LinkedIn at least. Tell me a little bit about your life growing up in Dallas, going to med school there, and then kind of how your career took you to different CEO jobs until where you got to today with Plus Therapeutics.
Yeah, you're right. I'm from Dallas originally, grew up in Texas, and then after medical school in Dallas, moved to California, lived there for 30 years, had a family there, had my dream job. I was an academic surgeon at UCLA and left on a 1-year sabbatical to start a biotech company with the support of my boss, who was the chair of surgery, and that was about 25 years ago. And so one thing led to another, and ended up giving away my academic job and going full into business, and have developed in my academic and business career a number of new med tech, pharma, surgical technologies for fetuses, babies still in the womb, pediatric patients and adult patients. And then the most recent iteration of that is developing targeted radiotherapeutics for patients that have some of the worst cancers out there, which are cancers of the central nervous system.
So I've had a bunch of biotech CEOs on the podcast, and they usually do have some sort of start in academia, kind of like you were saying, UCLA. Tell me about just kind of your mindset of when you went on the sabbatical of, okay, first, were you just going on it just because you said you needed a break? Did you have an idea that you always wanted to start a business or go into researching a specific thing? You just didn't have time with your current day job? Like, what was your thought process going into that? Did you have a plan, or did they kind of just fall upon you?
So, yeah, it's a little bit generous to say it was a sabbatical. It sounds like it was sort of planned and well thought through. That was absolutely not the case. So, way back in maybe 1999, 2000, I was the most famous man in America for about 5 minutes one evening on a slow news night. We had my group at UCLA cloned a first stem cell found in human adipose tissue, which has a lot of implications for all sorts of things. And that fame that happened really that was completely unplanned turned into a company and turned into some initial funding, and then turned into a public company with a lot of help from a lot of different people. And we were one of the first U.S. public stem cell companies. And that was, 25 years ago. So it really was sort of it was meant to be a temporary move into business. And I had my dream job in academia. But I realized once you take people's capital, you really, you're obligated to see it through, and you can't really go back. So it's kind of it's a one-way door. It's very difficult to go back the other way, but it's been great. I've learned a lot. And, I think what we have right now in terms of being able to, I think, safely and likely improve the survival for some of the worst cancers out there is the most exciting thing I've done in my career.
So to kind of follow up on that before we get into Plus Therapeutics, so you're working on the cutting edge of stem cell research, and then all of a sudden you become the most famous man in America, as you said, for 5 seconds, and people just start throwing capital at you saying, privatize this, make sure it monetizes. What was the process there? Did you right away know, okay, this is a traditional business plan? Where did you get up to speed? You'd been researching such a specific thing that now you have to be also like a business expert and an expert of running a publicly traded company. Like where did that knowledge come from, and kind of like who was your mentors along the way? Were you going to night school, or kind of how did that all come about?
Well, so yeah, it was really on the job training. So um, I think it's a little too much to say people were throwing money at us, but there was certainly capital out there. It was a different time in the markets. Now it's a little bit tougher on the capital side, but I think early on in with a business plan in hand and out trying to capitalize the company, I realized that a physician going against financial types, I was mismatched, and I could I could do my part academically and scientifically, medically, very well, I thought, but trying to match that to operating a business, financing a business, I was really um, not not suitably equipped to be able to do that. So I learned from my previous CEO and others that in terms of our board members and then bankers and other things. And then finally, I decided to get an MBA at UCLA. And what I found with the MBA is it's mostly social science. There's not a whole lot you learn that's any particular uh, uh, secrets of the world. Medicine, you learn a lot of really secret stuff that's unique and will never change. And human anatomy and biology, they're fixed. But business is mostly social science. But you learn a lot of terminology, and so you can if you can compete head to head on the terminology, then I felt like that's pretty much 90% of the way there. And just in terms of being able to sit across the table from someone and find common ground. It's fair for your shareholders and fair for both parties involved.
So you work at UCLA, then work at a few different biotech type companies, and then tell me about you first hearing about the Plus Therapeutics job, the idea. What first attracted you to the idea, and like where did you think you could add value?
Yeah, so there are a couple of things that are going on at the same time. One is our previous stem cell technology was there were there were folks that are interested in acquiring it, and we felt like they were better positioned to take it to the next step. Um, and so we divested that technology, and with that capital, we had some flexibility about where we might go directionally, and we had some cancer technology. But I became very intrigued in talking to a number of people, including one of our medical collaborators today, who's a well-known neuro-oncologist, that targeted radiotherapeutics had the best chance of prolonging survival for glioblastoma and other brain cancers that have very horrible prognosis. And it was a simple idea, like a lot of new businesses are. Simple at first, they just get harder later. But the idea was simple, that radiation is the best killer of cancer cells in the brain. The problem is you can't get enough radiation to it to really get meaningful improvements in survival. It's something that's called the low therapeutic index. You just can't dial up the radiation high enough to make a material difference in the patient's outcome without getting complications. Targeted radiotherapeutics allowed that, and so we've done 5 business transactions to acquire technologies over the last 5 or 6 years, including that initial technology for glioblastoma that at the time was funded by the National Cancer Institute that came out of the University of Texas. And we kind of added to that and built a portfolio not only of therapeutics, so drugs that can deliver high doses of radiation to the brain and spinal cord for patients with bad cancers, but also the best diagnostic of those. We realized that because there weren't therapeutics, in a way, the diagnostics hadn't really caught up. So diagnostic technology was as bad as the therapeutic technology. So we acquired a kind of a best-in-class diagnostic for those kinds of cancers. And now we're moving them forward in parallel, and we're trying to realize the synergies between the two. That's a longer story, but that's kind of how we got from that sort of simple concept to where we are today.
So yeah, you're gonna have to break this down for me, the layman.
Yeah.
So you have on one side of your business, you have like kind of a a diagnostic tool that can tell you exactly down to a certain measurement exactly where the cancer is in the brain. And then you have another kind of like syringe type tool that administers and makes sure that the chemotherapy type radiation goes straight to only that point in the brain.
Let me kind of try to simplify it and kind of break it down into two parts. So first of all, you mentioned the diagnostic. Let me start with there. So when I was in medical school 40 plus years ago, one of the ways you diagnose cancer in and around the brain is you take some fluid from around the brain. It's called a lumbar puncture. You've probably heard of it. It's very, very, very common procedure. It's done in the clinic with a small needle in the lower part of the back, and you pull out clear fluid, and then you assess it. But the assessments that you do on the fluid are incredibly crude. They've been around for decades, and they don't really quantify anything or tell you what kind of cancer it is or tell you even more complex things about what are the genetics of the cancer and how does that drive our selection of therapeutics, for example. So the technology prior to our diagnostic was you would take cells, spin them down in a centrifuge, smear them on a glass slide, put some stain on it, and then you would say one of three things: I think there's cancer there, or I don't think there's cancer there, or maybe. That was the test, and that's still the standard of care today, quite honestly. So we have a test that could tell you how many cells there are per cc or ml of fluid. So you get down to one cell per mil of fluid, which is incredibly sensitive. It can tell you what kind of cancer it is, confirming the diagnosis, and almost as importantly, it can rule out the cancer. So if you're a woman with breast cancer that might have some neurologic symptoms, and you're worried that the cancer is now in your brain or around the brain, you could take some fluid and we can tell that woman, no, you don't have it, which is an incredible relief. And you don't have to send her down a treatment pathway that's unnecessary. And we can do that very quickly and very reliably. So we could tell, is there tumor there? Is there a target for upon which to base your therapeutic? Because some therapies work better than others, depending on the kind of breast cancer, lung cancer, so forth that's gone to the brain. And then we can monitor the treatment. So if the tumor cells are going down, hey, your therapy's working, right? If tumor cells aren't going down or they're going up, then by golly, what you're doing is not working and you need to try something else. So it's that level of detail you get from this diagnostic technology that we have, and it's called See Inside, like a pun on See Inside, but it's actually CNSIDE. So See Inside Therapeutics, and that's a wholly owned subsidiary, and we'll be launching that test here in the next few months. So that's a diagnostic. But what it does for our therapeutic is it makes it much easier to diagnose cancer around the brain, so you can treat it early and you can treat it properly and you can follow it. So now if you kind of switch over to the therapeutic side, our therapeutic, our lead therapeutic, I'll give you a little bit of a hot off the press. The FDA has approved the name of the drug. It'll be called Rheobic. That'll be the commercial name. And so Rheobic contains a chelated encapsulated form, which means it's radiation inside of like thousands of radiation isotopes in a ball of lipid, and then that ball of lipid goes into the fluid around the brain and irradiates the areas that have the tumor, but doesn't affect the normal brain tissue. That's big picture how it works. But by having the diagnostic married to the therapeutic, we can increase the opportunity to use our drug on these patients by 2 to 4 times because it's so poorly diagnosed. And so, since kind of going back to what I mentioned in the beginning, is that cancers of the central nervous system are incredibly radiosensitive. They're sensitive to radiation. You just can't get enough. So our drug allows us to safely get maybe up to 20 times the amount of radiation that you can get with standard medical radiation that is delivered today in essentially every hospital in the US. So we're dialing up the radiation up to 20 times, but we're doing it on patients that we absolutely know what their tumor is, how much tumor bulk they have, and then we can follow it over time. So the diagnostic and the therapeutic really work synergistically to increase the opportunity to diagnose and then treat these patients. And in most cases for these diseases, nothing's approved by the FDA. So it's not like we're going in with a Me Too drug. This is not only a new type of drug, but it's for a group of patients that really don't have good therapeutic options today.
A couple questions about the FDA process. The first one, since it's hot off the press, and you don't have to spend too much time on this, but what's the deal with the drug naming process? They seem like they're the least marketable names of all time. Whenever I see a commercial for a pharmaceutical or something like that, is there something purposeful about that where they have to be like non-marketable, or how do you go about even getting approved naming wise to the FDA before you even get approved for everything else?
Yeah, it's not just a bunch of guys that watch a lot of golf commercials that sit around and think up these crazy names. It's an expensive process. It requires a lot of consulting and global research. So they take into account physicians' prescribing practice because they want to make sure, from a safety perspective, it doesn't. When you write it out, it doesn't look like another drug that's completely unrelated, for example. And then you have to look at all sorts of languages. You can't just look at English. You have to look at Portuguese, and you have to look at how things are translated. So not only how it's written, but how it's said. So if the doctor's telling, a pharmacist or another doctor, hey, prescribe this drug, if it sounds like another drug, then there's opportunity for confusion. So there's a tremendous amount of focus group testing and analysis linguistically to come up with the right name. But when you distill it back, the essence of the name starts out with what is the drug and what does it do? And so Rayobic, RE, is the radioisotope that we use in our drug. It's rhenium, and RE is the symbol for that. So there's some things in it that really describe the drug, but then it gets morphed over time, so it's safe, and that's why the FDA has to sign off on it because of the potential safety issues.
No, it definitely seems like something that would be well suited for AI. With you saying, hey, here's what my drug does. Hey, make sure it doesn't sound like any other drug. Make sure it's pronounced easy in every language and stuff. That's interesting. Okay,
that's true.
The more serious side of the FDA side of things. So as you're getting approval on one side for the diagnostics and on the other side for the therapeutics, do you have any say in saying like, hey, this diagnostic is meant to be used with this therapeutic, so like don't use it on its own, or vice versa? Or do they get approved together as they go through it, or are they approved separately? How does that work?
Yeah, it's a great question, and sounds like you know your way around the FDA a little bit. So, it's actually it's really germane. So, you can have diagnostics they're called companion diagnostics, and so they're really fundamentally married to the therapeutic. Oftentimes, they're very specific, and they look at one gene or one feature of the tumor, and then they marry it with a very specific type of therapy, and so their approval process is sort of in parallel, and the FDA approves the companion diagnostic. On the other hand, there are general diagnostics, and the diagnostic I mentioned, our CNSide diagnostic platform, it has elements of being specific, but its major importance is in its generality. It can function as a screening test for cancer in and around the brain for patients that have very common cancers like melanoma, lung cancer, breast cancer. Those tend to you don't tend to die of the cancer that's in your breast or in your lung. You die because it metastasizes to organs and tissues that when once there, you can't eradicate it. And the brain is the prime one because there's something called the blood-brain barrier. Believe it or not, there's this biologic barrier that keeps stuff that's circulating in your blood from getting into your brain to protect your brain, but it also keeps 98% of drugs out. So so when cancer pops up in and around your brain, the prognosis is very bad and the treatment options are very small. So our diagnostic actually has different pathways than being a companion to a therapeutic. It actually is a screening tool. It's a therapeutic monitoring tool. It can rule out the disease as well as rule in the disease, and it can suggest therapeutic monitoring. So that pathway is different. And in fact, there are a couple different pathways. One goes through the FDA, and that's our long-term, that's our intention. But you don't need that. There's actually another pathway that goes through CMS. CMS is basically Medicare. It's under Health and Human Services. But there's a regulatory pathway that's been out there for decades called the laboratory developed test. And the bar is much lower than FDA. And that's what allow us to move much more quickly and get that into commercial sales because that bar is lower. So we're actually pursuing the laboratory developed test pathway first, which gets it into the market. And then over time, we'll then be getting FDA approval that would help us kind of build the story and the notoriety and the kind of regulatory girth behind it. And there are a lot of reasons to do that. But it's very different than that companion diagnostic that's done in marriage with the therapeutic.
And I talked to some companies kind of in your position who it seems like they may, because of just the way the market is, have a path to actually like manufacturing the product itself. It doesn't seem like that makes as much sense maybe for you all. What's the plan for scaling this out once it gets approved? Is it to license it out to someone else? Is it to use, say, like the diagnostics and see if it can get approved for 50 other cancers that it can maybe help with? And like, how do you plan on, say, scaling over the next 5 years if everything goes to plan?
Yeah, let me break that down again into two parts. So for the diagnostic, one of the great things about the diagnostic is it's done centrally. So we have a diagnostic laboratory in Houston that's right in between Hobby and George Bush Airport near MD Anderson. And so specimens throughout the US come to one place. All this facility does is testing on fluid around the brain for the reasons I mentioned before. So scaling is really just a matter of expanding the footprint, increasing the number of people, and then there's some sort of back end scaling and so forth. And there's some devices that go into it that make the test less expensive and faster. And so those are relatively easy to scale. So that's pretty straightforward. It's really it's time, materials, and space that allows scalability, but it's relatively straightforward. On the drug side, completely different. Radiotherapeutic drugs, or drugs that deliver radiation, are different than most other drugs for one principal reason, and that's the shelf life is very, very short. So a radioisotope, by its very nature, decays. So you put it on the shelf, every period of time, the amount of radiation delivers gets
a half-life for those types
it's
of
a
drugs?
half-life. Yeah.
Okay.
Yeah, exactly. Yeah, no, it's exactly. So it's a half-life. It's a radiotherapeutic, but it's the same thing in shelf life. So you can't just sit it somewhere and pull it off 6 months later. You really have to make it in real time. So you have to build a supply chain that allows for the nurse, the doctor, the OR scheduler, whoever's taking care of the patient. They phone the company. We have all of the intermediates made, which are long shelf life. And then we irradiate the starting material. That's done overnight. And then the drug is made the next night. And then it's shipped out. And then it can sit on the shelf at the hospital for maybe 4 days. It's got to be used within that sort of very compressed time frame. But it's really made to order. But that supply chain, you have to scale the radiation services to make the radioisotope. You've got a scale for the different intermediates based on shelf life and availability and so forth. And then you need manufacturing scalability throughout the supply chain to be able to make it in real time. And you need backup. These patients can't wait oftentimes. You can't just say, "I'll come back in 6 months." You got to treat them that week or the next week or very soon thereafter. So you need a supply chain that can react very quickly and is redundant. So if part of the supply chain fails, which you have weather, we might need to treat a patient, when there's an active tornado or some other weather system that affects, flights in and out because it's overnighted generally with flights. So you have to build in some level of redundancy there. So it gets very different, almost kind of different ends of the spectrum in a way.
Yeah, for sure. And can you use like a traditional like FedEx overnight courier? Is there like a special like radioactive type courier that handles these types of drugs?
You can use standard, and it really depends on the radioisotope. There's some that are, they put out lots of radiation, and they send out the radiation very long lengths, and so they need very lead, for example, or something that will absorb that radiation. Ours happens to be very easy to ship. The length of the radiation on average is only about 2 millimeters. So if it's in a syringe, arguably it won't even penetrate the wall of the syringe because the path length is so low. But the path length is really critical in being able to target it to the tumor and then not get normal brain, for example, or normal tissue that you don't want to hit. You just want to hit the tumor. So the path length becomes very important, not only in targeting, but also in shipping.
Super interesting. Well, Marc, as we wrap up here, kind of ending question for a lot of people is just what gets you up in the morning? What gets you excited about the company? Besides just the helping people, do you have any other things on the sidelines that you're looking to go after or anything? And then kind of to add on to that, what's one thing you would tell yourself working back at UCLA, like medical wise, of being like, hey man, you really should look into isotope so and so, or what would you tell yourself that like, hey, this could have really sped up things a lot quicker?
So to the first question. So the other day, I had a little free time, and I was thinking, okay, look, I have high confidence that our diagnostic and our therapeutics are going to be game changers. How do I convey that in a very simple idea in terms of what's the value? And so I got out a little notepad and made a few assumptions, and that I thought were reasonable to conservative. And based on my calculation, if our diagnostic is approved and our therapeutic is approved, and both are on the market, we can realistically add 150 million days per year of life to this very small group of patients.
Wow.
Every year. So, small increments approval because of better diagnostics, and even better increments of approval because of the therapeutic based on the data and the way it looks right now. And so that now I sort of have this number, 150 million. I mean, that's reasonable. And you think about, okay, who is that in that 150 million? Maybe there are people that are going to live an extra year. Maybe there are people who live 6 months, but they have better